Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall continue discussing how our cells regulate metabolism, and we will look at the availability of cofactors as a way to regulate metabolism. As discussed in the previous videos, we could change metabolism in the cell either by changing, for example, the function of existing enzymes or the amount of the enzymes present in a cell. In the last video, we discussed how we can change the functions of existing enzymes by placing them in various compartments. And in this video, we'll see how the availability of cofactors could affect the function of an enzyme and therefore affect or regulate metabolism. There are many proteins, including enzymes, that cannot work by themselves. They require the help of cofactors, which are non-protein molecules that are required for the functions of certain proteins. Cofactors could be either inorganic molecules, such as the metal ions, examples magnesium, iron, copper, zinc, manganese, and so on. Or cofactors could be organic molecules, and examples of this include heme that we discuss under myoglobin, hemoglobin, and the cytochromes. And then we have various coenzymes, and many of these coenzymes you can see are derived from the B vitamin family. For example, FAD, the oxidized form, and FADH2, the reduced form, are derived from the vitamin riboflavin, which is vitamin B2. NAD and NADH, as well as NADP and NADPH, are derived from niacin, also known as nicotinic acid, which is vitamin B3. Coenzyme A is derived from pantothenic acid, which is B5. TPP, or thiamine pyrophosphate, is derived from thiamine, which is B1. Then we have biotin, another coenzyme, derived from B7. Pyridoxine B6 is the vitamin that we use to synthesize pyridoxal phosphate, or PLP. THF is derived from folic acid, or B9. And then we have cyanocobalamin, which is vitamin B12. In previous videos, we discussed how these coenzymes help certain enzymes carry out or catalyze certain types of reactions. For example, redox reactions. We have certain enzymes that require FAD or NAD or NADP. Enzymes that catalyze acyl transfer require coenzyme A. And then there are certain enzymes that carry out decarboxylation reactions as well as transketolation reactions. They require thiamine pyrophosphate, TPP. Then we have certain enzymes that carry out carboxylation reactions, and they require biotin. And then we have enzymes that carry out transamination reactions that require pyridoxal phosphate. Enzymes that carry out the transfer of one-carbon chemicals require tetrahydrofolic acid. So if these enzymes require coenzymes to function, it is therefore not surprising that if a certain coenzyme is not available in sufficient amounts, then we would affect the function of the enzyme and therefore the rate of the metabolic pathway that the enzyme is involved in. Let us use one example to show this concept of how the availability of cofactors we'll use the example of NAD. So how the availability of cofactors can affect the function of the enzyme and therefore regulate the rate of the metabolic pathway. Many enzymes that we talked about in previous videos require NAD as a coenzyme. For example, in video 4.1c, we discussed the enzyme glycerol 3-phosphate dehydrogenase, and this enzyme catalyzes, you can see the reaction between glycerol 3-phosphate, phosphate, and requires NAD to give us 1,3-bisphosphoglycerate, and the oxidized NAD becomes reduced NAD in the form of NADH. So these kinds of enzymes, they will have a domain, a part of the enzyme that will bind the coenzyme, in this case NAD, and then they will have the catalytic domain, the active site that will catalyze the reaction, in this case the reaction between phosphate and glycerol 3-phosphate to give you 1,3-bisphosphoglycerate. So let's look at the first example. In this case, we have a cell in a high energy state, which means this cell has a lot of ATP present in the cell. So when the concentration of ATP is very high in the cell, that ATP is going to inhibit complex 4 of the electron transport chain. Complex 4 is also called cytochrome C oxidase. So the inhibition of complex 4 will decrease the transfer of hydrogen from NADH and FADH2 to oxygen. So NADH and FADH2 will not be able to give their hydrogens to oxygen. That will lead to an accumulation of NADH and FADH2, which are the reduced forms of the coenzyme NAD and FAD. Now, the high amount of NADH present under these conditions are going to go and inhibit certain enzymes. For example, it will inhibit pyruvate dehydrogenase complex, the PDH reaction here. So this enzyme will be inhibited by the high concentration of reduced coenzyme NADH. The high NADH concentration will also go and inhibit isocitrate dehydrogenase. This is the enzyme that converts isocitrate to alpha-ketoglutarate. So this enzyme will be inhibited. And when this enzyme gets inhibited, citrate amounts accumulate. The increased citrate amount will then go and inhibit phosphofructokinase. This is a key enzyme of glycolysis, and that will even slow down glycolysis, because now this enzyme is inhibited, it is going to slow down glycolysis. The high NADH, this reduced form of coenzyme, will also inhibit alpha-ketoglutarate dehydrogenase. This reaction here, where alpha-ketoglutarate is converted to succinyl-CoA in the Krebs cycle. Note that this enzyme here, malate dehydrogenase, this enzyme is not inhibited. So the conversion of malate to oxaloacetate is not inhibited because this is a reversible reaction. The delta G is close to 0, and therefore this will not affect the rate of the Krebs cycle. The high amounts of the reduced NADH will also inhibit citrate synthase. So this enzyme here that has catalyzes the reaction between acetyl-CoA and oxaloacetate to form citrate. So this enzyme will also be inhibited. So with the inhibition of these enzymes of the Krebs cycle by NADH, the Krebs cycle will slow down. There will be a slower oxidation of acetyl-CoA into carbon dioxide. There will be decreased amounts of NADH and FADH2, and eventually the energy state of the cell is lowered back to normal values. So this is one example by which coenzymes, in this case NADH, can affect the rate of metabolic pathways. Let's look at another example, and in this case, we'll look at the NAD deficiency due to ethanol metabolism. So when we drink ethanol, ethanol is metabolized to a minor extent by cytochrome P450. So this reaction here. But the major pathway by which alcohol is metabolized is by the enzyme alcohol dehydrogenase, which converts ethanol into acetaldehyde, and NAD is required, and the NAD becomes NADH. So basically, NAD is used up to metabolize ethanol, and NADH will accumulate. So that will lead basically to a NAD deficiency. Now the acetaldehyde will enter the mitochondria, and they will be converted to acetic acid. And once again, the NAD is used up, and we get NADH form. And this reaction is catalyzed by the enzyme acetaldehyde dehydrogenase. So once again, NAD is used up, so there will be NAD deficiency in the mitochondria, as well as NAD deficiency in the cytoplasm. So this NAD deficiency in the cytoplasm, as well as the mitochondria of hepatocytes, the liver where most of the ethanol is metabolized, is going to affect all cytoplasmic and mitochondrial enzymes that require NAD as a coenzyme. Remember, NAD comes from a vitamin, niacin, and it's a vitamin niacin that we cannot synthesize. We humans cannot synthesize. So, and it's also very difficult. The pathways to make NAD are quite complex. It takes time to make NAD. So this can all lead to a deficiency of NAD, in this case in the hepatocytes. So this NAD deficiency is going to affect certain enzymes. For example, fatty acid breakdown, beta oxidation requires NAD, and without the NAD in the mitochondria, remember fatty acid oxidation occurs in the mitochondria. So without this breakdown of fatty acids in the mitochondria, what's going to happen is there's going to be increase in fatty acid synthesis, and that is going to lead to increase in triglyceride synthesis and accumulation. And this triglyceride accumulation in the liver is going to cause what we call as fatty liver. Now, under normal conditions, the liver gets lactate from various parts of the body, such as your skeletal muscles and your red blood cells. And then the hepatocytes will convert this lactate into pyruvate. And then this pyruvate is used by the liver to make glucose by gluconeogenesis. Now, the conversion of lactate to pyruvate, catalyzed by LDH, requires NAD. So this requires NAD. However, since the NAD is being used up for ethanol metabolism, there's going to be a NAD deficiency, so the liver will not be able to convert the lactate into pyruvate. And that NAD deficiency will therefore lead to lactate accumulation. And that lactate will spill out into the bloodstream, and that will cause lactic acid acidosis. So once again, we see how the availability of cofactor, in this case a coenzyme NAD, can significantly affect various metabolic pathways. In the next video, we shall look at another way of regulating the catalytic activity of existing enzymes, and this is allosteric regulation. So until the next video, once again, thank you very much for watching us and supporting this channel. Goodbye and have a wonderful day.